Hi, welcome back. I just wanted to take a moment to say thank you to Dr. Urich and the audience also for such wonderful questions. Just as a reminder, you can ask questions during any of the presentations by using the Q&A button in the lower left corner of your screen. Also, if there's a break between speakers or at any time, you can visit our Life Technologies branded virtual booth and chat with a representative there. I have the pleasure of introducing our second speaker today, Dr. Nikolai Khazanov, who earned his doctorate in bioinformatics from the University of Michigan. He worked on structural bioinformatics methods for computational drug design. And today he is speaking about the analysis of the Oncomine Knowledgebase to identify NFE2L2 pathway as a novel therapeutic opportunity in multiple cancer types. And now I'd like to turn the controls over to Dr. Khazanov for his presentation.
Hello. Thanks for tuning in. My name is Nikolai Khazanov. Today during my talk, I'll talk briefly about the analysis of Oncomine Knowledgebase to identify novel therapeutic opportunities in multiple cancer types. Specifically, I'm going to use an example of NFE2L2. So in contrast to Dr. who came before me and Dr. Eddy after me, I work on more on the bioinformatics side of things, developing methods and ways of analyzing our data and really leveraging the Oncomine Knowledgebase. So my talk will focus slightly on how our approach to ingesting and analyzing the data before we can make these insights into therapeutics from it. So bioinformatics is really at the core of what we do here within the Oncomine and Compendia services offerings. We work closely with the bioinformatics team works closely with the translational scientists whom you've heard speak before and will hear speak after me, who really help us extract insights from the data. The way that we approach our method analysis is to really enable us to identify novel therapeutics targets, identify the right patient population, and design well-informed diagnostics going into the future, giving the landscape of the cancer data that we've collected and analyzed. So our approach is to define a comprehensive catalog of driver aberrations, and that is what we can mine for all the biological and therapeutic inference. We focus on data types that are largely relevant to current clinical cancer research. That's mutations, fusions, copy number aberration, of course, expression data as well. And so NGS data has really been poised to accelerate the discovery of new target identification, especially of genomic markers of response, and the models to characterize these candidate drivers. So really to maximize it, we have to accurately assess the aberrations that are present in cancer data, really delineate the driver aberrations from those that occur by chance and perhaps don't have functional impact. Annotate them for potential clinical relevance and integrate them both by gene, pathway, and cancer type dimensions, so all the different ways that it's helpful to summarize the data. So a brief snapshot of the knowledge base, the Oncomine knowledge base, and how we approach it. We try to source all publicly available cancer data, again, within those data types and data sets that are applicable for answering questions about tumor targets or tumor populations. So gene expression is certainly the largest bolus of data since it's been accrued historically over many years, but we have significant sources of NGS data as well, such as mutation, fusion data, and other data types. The knowledge base feeds multiple efforts within our work. Some of that is the services work that you've heard Mary Ellen talk about, and you'll hear Sean talk about. But the knowledge base also provides data for our power tools, which is really a way for our customers and for our internal scientists to drill into specific data types to understand really what the source data is telling us about a specific cancer type or a specific target. So the knowledge base also supports the services works, as I've mentioned, and for that there's additional methods and additional data types that we pre-compute to enable answering the most basic questions that come up during either oncopredictor studies or target validation studies or other offerings that we perform through the services work. So the challenges for us, I really think, are also end up being our strength, since these are the areas that we've focused on and really have learned to handle over the years. So data, large-scale data collection and processing, you have to be able to get the latest data. You have to be able to assess the data sources that are out there and get access to that data and really bring it in systematically and repeatedly in order to maintain a current collection that's relevant for the current science. So different formats and analysis methods are obviously a function of scientific research, and thus it's important to identify both the methods that are, for example, consensus methods. If there's a method that works best, let's use it. If not, if there's no consensus, for example, what methods works, let's develop our own so we understand it. So we really strive to use common, clear methods and to clean up the data so that the data itself is not heterogeneous, and that's a major effort that I'll touch on briefly later. Speed. So as I mentioned, iteration is important, and with NGS data coming out in floods in recent years, you really have to pay attention to what new data sets are out there. And last but not least is certainly this exercise of identifying true driver events from methods that might not be mature yet, or might still be under investigation. So the identification of driver events is something that's a big advantage when it comes to mining a large heterogeneous data set that was compiled from multiple data sources and contains information on thousands of samples amongst hundreds of cancer types. And I'll touch on that briefly as well. So in general, when we are making a power tool, for example, that would allow one to integrate or to analyze NGS mutation data in depth, we follow a generic approach to getting the data, analyzing the data, and then presenting that data to a scientist. And so this is a general approach that I'll describe here, but I'll touch on how that applies to the different power tools in the latter slides once I get to my example of NFE2L2 and how that potential therapeutic target was characterized. So as I mentioned, we start with the biggest collection of genomic data, cancer genomic data we can find. We then utilize the best methods on it, and that can take a variety of forms. For example, for fusion analysis, when we started collecting RNA-seq for fusion data identification, there were no sources of fusion calls, so we had to create our own framework for computing fusion calls from RNA-seq data, and that involved the high-performance grid and many years of compute time that we were able to crunch through. Some problems require not only brute force computation, but manual curation and annotation, and In that department, we have tremendous resources in our curation team that help us standardize cancer types, for example. So if you can imagine grabbing mutation data from many different studies that use their own cancer type definitions, our curation team really helps us standardize that data and make it accessible to our analyses methods. So the resources to process and standardize the data is something that we put a lot of effort into. From there, we have now a somatic aberration database where things like mutations, fusions, copy number aberrations have all been standardized to a single format, so the data sets essentially don't matter. We can look at either pan-cancer or within cancer, however we want to slice this data. And from there, we really move on to the driver classification. So for the driver classification, again, there's probably as many methods as there are scientists for classifying a potential driver gene in cancer, for example. But we really work with our internal scientists, such as Dr. Eric and Dr. Eddy, to understand the questions that our customers are asking of the data and be able to provide driver classifications that are consistent with the current definition or current consensus in the field. So for that, you have to use gold standards, and gold standards have to be vetted by cancer biologists who understand the therapeutic and the functional sides of cancer biology and can really guide knowledge extraction in a very supervised but powerful way. Of course, whenever you do a classification, you have to understand why you're classifying something, and so we really strive to develop algorithms that stem from biology and not just simple, not maximizing a separation between two classes or something like that. Of course, we retain all the original data so that we can assess whether the classification of drivers and analysis of drivers have really gave us an advantage over analyzing the entire data set. So that was a general approach that we take for a lot of our analyses and tools. I'm going to quickly touch on a specific analysis that we do several times a year, and that supports a lot of work that our services team does to characterize and identify novel targets. So this is the integrative analysis, and the concept here is to take all the NGS data that we have available and perform really common analyses that are common questions that a scientist wants to ask of a tumor data set, and have those pre-computed for easy identification of significant events. So from simple things like the frequency of genomic aberrations, how frequent is this mutation in this cancer type, all the way through to correlation of the DNA and RNA levels in a gene given a mutant or wild type status, or given a fusion or wild type status, for example, all the way through to outcome data. So for many patients, retrospective outcome data is available now in the public data sets. So by using that data, you can calculate survival curves and assess whether there's a significant difference of survival in at least using this retrospective data, if there's a significant difference in survival amongst different patient populations that are aberrant or wild type. When these analyses are performed on all the data in a hypothesis sort of agnostic approach, it really leads to powerful methods of extracting knowledge from that data. And here's an example. So this is for one of our recent integrative analysis builds. We start with just under 9,000 clinical tumor samples. This is data from clinical tumor samples. These samples have been characterized for mutations, fusions, copy number, and many of them have outcome data. They also have expression data. What you see in the chart in the upper right corner is that if we were to define apologies. if we were to define genomic events based on any mutation or any fusion or any copy number we observed, we would end up with as many events as their genes. So the top line there of any gene aberration just signifies that a gene was aberrant in some way, whether it's a mutation, fusion, or copy number. When you add that up for these 9,000 samples, almost every gene is aberrant. So really what you want to do is focus on aberrations that look like driver aberrations. Now these might not be functionally validated, obviously, but these employ the heuristics that we've developed by looking at known driver genes, whether oncogenes or tumor suppressor genes, and classifying the rest of the data in accordance with those heuristics. So we can see that when applying driver definitions, the number of events for us to look at reduces to 5% of the original data. So it really helps us zoom in on those aberrations that we think will be important as either therapeutic targets or for other purposes. The pie chart on the left shows the fact that care is taken to combine the data appropriately. So for about 4,000 of these 9,000 samples, we actually have data on multiple data types for a single patient, or for a single tumor sample. So in this case, a tumor sample is measured for mutation, fusion, copy number, and expression, and it has outcome data. So those are obviously more powerful for some of these integrative analysis. So integrative analysis is just one of these tools that we put together to look at NGS data. We also have NGS-specific tools for expression, fusion, and mutations, as well as copy number and some legacy expression data tools. Overall, these represent thousands of tumor samples available for analysis, and contain some sort of classification schemes to identify potentially important driver events. Now, one way to approach a target discovery exercise with such a data set is to have a consensus score that identifies genes that are identified as drivers or significantly aberrant in multiple data sets. Our scientists have put together just such a scheme using a gold standard set of genes. So these are gold standard. These are genes that are known in literature and have been shown to play a role in cancer incidence or cancer progression. So by building a consensus score, we can generate this spectrum of gene ranks on the spectrum for a potential what looks like an oncogene to a tumor suppressor gene, and look at the interesting genes that pop up that are obviously not the well-known ones that were included in the gold standard. So now, for the rest of the talk, I'll want to take you through an example of such a gene that we discovered. And although there is literature describing NFE2L2 and its potential role in cancer, the slides will show how our tools and our data can help tell a biological story around this gene, even if you didn't really know all that was published in the literature ahead of time. And that is indeed what we did, what our team did here. And this is not solely my work. This is work with help of my services team colleagues. So just a brief primer on the biology of NFE2L2 and the protein NRF2 that it encodes. So NRF2, under normal conditions, is actually usually degraded. It's targeted for degradation by KEAP1, with which it forms a protein-protein interaction. Under conditions of oxidative stress, though, this interaction is broken due to stabilization of KEAP1. And what happens is NRF translocates naturally to the nucleus, where it starts transcribing many genes, including antioxidants, enzymes that alleviate the stress on the cell. So it essentially allows cells to survive stressful conditions. So this is the biology of the NRF or NFE2L2 gene that we saw pop up as one of these interesting potential driver genes. So if if we look at within our NGS mutation browser, we summarize mutations, somatic mutations from over 7,000 samples, we can actually see that there's quite a prevalence for the NFE2L2 and KEAP1 mutations in multiple cancer types. Sometimes the literature could identify a potentially important cancer type by really looking at a pan-cancer view like this. You can get a sense of potential clinical populations within other cancer types with similar mutations. You can see from this bar chart that, for example, in squamous cell lung carcinoma, there's almost a 13% incidence of NFE2L2 mutations. You can also see that the mutations are colored by their hotspot or deleterious status, and that status is part of the classification scheme that allows us to say, hey, these are recurrent mutations in this gene. They're probably more important than passenger mutations and ones that aren't recurrent. And you can see that by looking at the transcript level. This is also from our NGS mutation browser. It shows the NFE2L2 amino acid position on the y-axis and count of samples aberrant on the y-axis. You can see that the recurrent mutations, ones that pile up at a single position and are observed across multiple cancer types, are colored in solid, and those are the hotspot mutations. Those are the hotspots that, since evolution have chosen them to recur in a single position, are assumed to be functionally interesting. And you can see that the recurrent mutations do in fact occur in the KEAP1 binding domain, and it's when this binding is interrupted that NFE2L2 does its job of preventing or alleviating cell stress. You can also see that there's a lot of mutations that are not colored because they haven't been found to be recurrent and those not considered to be potential driver mutations. KEAP1 has a similar view in the NGS browser, NGS mutations browser, with recurrent mutations in the NFE2L2 binding domain. So both genes exhibit recurrent mutations within cancer, within tumor samples of multiple tumor types in their binding domains. More interesting is that if you extract the tumor samples that have these hotspot driver mutations, and you look for co-occurrence of the KEAP1 and NFE2L2 mutations, you see there's high mutual exclusivity, high degree of mutual exclusivity. Most of the NFE2L2 mutations are hotspots, while the KEAP1s are largely deleterious mutations, so frame shifts and the like. This mutual exclusivity implies a functional relationship between the two roles, which we know is true from the biology of the two genes. So it only takes a hit on one of their binding sites to prevent the protein-protein binding. Since we've looked at a range of tools to identify NFE2L2 as a driver, it had significant results within our outcome analyses. And in this case, it was in head and neck carcinoma that we saw poor outcome associated with NFE2L2 recurrent mutations. That's not really intuitive at first, but the reason why it happens is because NFE2L2 actually helps cancer overcome some of the stress from chemotherapy. So once the patient undergoes treatment, if you have an NFE2L2 recurrent mutation, it's actually likely that it'll confer some resistance against the chemotherapy and help the tumor survive. So this identifies that a potential therapeutic opportunity within a retrospective tumor population here. And again, systematic analysis of outcome across all the cancer types and all available data might identify cancer types that have not previously reported this association with outcome. And this is actually a screenshot from the Integrative analysis browser that I mentioned before. So this is a pre-computer significant analysis that we can observe. Moreover, if you reach into the expression data in our knowledge base, in the Oncomine Knowledgebase, you can see that some of the top genes that are overexpressed in NFE2L2 mutant or KEAP1 mutant squamous carcinoma samples are actually targets of NFE2L2 transcription. So these are the genes that help combat oxidative stress. Conversely, if you look at the activation signature of NFE2L2, there are and you look, so this is a snapshot from our Oncomine Concepts Edition tool, which summarizes the expression data. If you look at the enrichment of the genes in NFE2L2 activation signature, you can actually see enrichment for cell lines with chemotherapy resistance. So this shows in the model system view how chemotherapy resistance is evidence of these mutations. So that data help us characterize a potentially novel therapeutic target. There's since we've also collect data on cell lines and cell line mutations, we can identify model organisms to then pursue the characterization of this novel target. So in this case, what I'm showing in the top plot here is NFE2L2 and KEAP1 mutations in cell lines that were annotated with the classification that was derived from tumor samples. So the solid dots are mutations that are hotspot recurrent mutations in tumor specimens, in clinical tumor specimens, and they've been overlaid on top of the cell line data. So you can identify then cell lines that have these recurrent mutations that were identified in clinical samples, and given data on cell line sensitivity to knockdown of various genes, which we have in our Oncomine RNAi browser, we can then identify a cell line that's actually sensitive to knockdown on NFE2L2 and could be potentially used as a model to characterize the gene further. So this kind of shows how the biological story for the NFE2L2 KEAP1 interaction and the potential therapeutic importance of NFE2L2 can be gleaned from this massive database of genomic and continuous data collected sort of from publicly available tumor data. This brings me to the end of my part of the talk, so I'd like to thank my colleagues, all of whom have contributed greatly to all the bits and pieces of this work, and I'll leave it open to questions. Thank you very much for your time.